MASH impacts approximately 5% of the population globally. The FDA approval of resmetirom introduced the first pharmacological therapy for patients with MASH with moderate to advanced fibrosis of stages F2 or F3. Wegovy has also been recently approved with a similar indication label. Imaging-based diagnostics and blood-based biomarkers are widely used in clinics to non-invasively determine eligibility for treatment. While non-invasive guidelines mark a step forward in patient management, there's a stark reality of applying these criteria to such a large patient pool. For example, using criteria CAP and liver stiffness via transient elastography, combined with ALT levels exceeding 17 U per liter, a recent study showed that between 2.3 to 8.3 million adults in the United States may be eligible for resmetirom treatment. New therapies mark the beginning of a new era in MASH management, but they also underscore the pressing need to ensure precise patient identification and optimal treatment selection. Alongside resmetirom, other recently approved drugs, such as Wegovy, originally developed for obesity management, are also entering the landscape. Together, these therapies mark the beginning of a new era in MASH management, but they also underscore the pressing need to ensure precise patient identification and optimal treatment selection in this rapidly evolving field. The main challenge with NITs is their limited sensitivity and specificity for detecting MASH with moderate to advanced fibrosis F2 to F3. A high inaccuracy rate between NITs and liver biopsies in prescribing resmetirom has been reported with underdiagnosis of over 60% of biopsy-eligible patients and simultaneously overdiagnosis of approximately 40% of those who did not meet histologic criteria. Furthermore, discordance among non-invasive tests, particularly between serum biomarkers and imaging modalities, is a recognized limitation, often leading to conflicting results, especially at intermediate fibrosis stages, F2 to F3. This highlights that the transition from histology-based clinical trials to real-world practice based solely on current NITs will have tremendous impact on accuracy and economic impact. For this reason, liver biopsy analysis remains essential, at least in selected cases. But standard histology also has well-recognized drawbacks. Inter-reader variability can lead to inconsistent staging between pathologists, and reliance on an ordinal, semi-quantitative scoring system limits precision. Together, the limitations of both NITs and conventional histology may lead to overtreatment and misdiagnosis of MASH. With high costs and the potential for overprescription at stake, accurate and reproducible patient identification has become critical in the era of FDA-approved therapies. We know that biopsies are still the gold standard for diagnosing MASH and staging fibrosis, so how can we most reliably achieve fibrosis assessment to accurately identify patients eligible for treatment? HistoIndex has pioneered a novel approach to assess fibrosis in biopsies. Based on second harmonic generation, SHG, HistoIndex has developed an advanced imaging technology, which allows the visualization of fine fibrillar collagen in tissues with exceptional clarity, without the need for stains or dyes, in a completely non-destructive manner. In this slide, you can visualize the difference in collagen fibril detection by SHG and Masson's trichrome on the same or adjacent sections of two different liver biopsies. SHG relies on intrinsic optical properties and avoids any stains, resulting in cleaner, higher contrast images of collagen, with reduced background and scattering, which you can clearly see in these examples with details which can be overlooked by traditional methods. In these instances, you can see images taken from two biopsy regions, which has been processed first with SHG on the right, then stained with Masson's trichrome on the left side. In the SHG images, we can distinctly observe fine fibrillar collagen and bridging fibrosis, in contrast to conventional staining and imaging approaches. SHG can detect collagen pixels down to a resolution of 0.39 micrometers per pixel to adequately resolve subcellular features like collagen strands and nuclei. The Genesis 200, HistoIndex's proprietary tissue scanning system, works based on the phenomenon of second harmonic generation, or SHG. To provide a stain-free assessment of fibrosis, unstained FFPE tissue sections are placed inside the Genesis 200 and laser-excited at specific wavelengths. SHG photons of half the wavelength of the original source are exclusively produced from interactions with non-centrosymmetric structures within tissue, notably fibrillar collagen. Simultaneously, two-photon excitation fluorescence TPEF microscopy detects cellular morphology. The intensity and spatial distribution of the SHG signals can therefore accurately map the extent and location of fibrosis as rendered in this example. The high-quality label-free images generated through SHG/TPEF imaging form the foundation for different types quantitative assessments, including the AI-based algorithm QFibrosis, HistoIndex's proprietary tool for liver fibrosis quantification in patients with MASH. The QFibrosis AI model has been trained on expert hepatopathologists' scoring of several MASH biopsies and is calibrated to generate fibrosis scores on a continuous scale. To date, scanning and QFibrosis scoring has been done by HistoIndex for thousands of human samples from MASH clinical trials, including the Phase 3 resmetirom MAESTRO-NASH trial for drug efficacy measurements. In multiple clinical trials, QFibrosis has allowed the detection of fibrosis regression, which cannot be detected otherwise by standard scoring systems because of the ability to quantify fibrosis on a continuous, nuanced scale. In different Phase 2 and 3 trials, including in this Phase 3 resmetirom trial, when the trial biopsies at baseline and end of treatment are analyzed by QFibrosis, the proportion of patients showing regression of fibrosis improved with the use of QFibrosis, lending confidence in drug efficacy. As with clinical trials, where conventional histologic assessments endpoints are limited by the reliability of the scoring systems, so too in routine patient management, where biopsies are taken, autonomously calculating a continuous QFibrosis score for a MASH biopsy would translate to consistent and sensitive fibrosis scores, importantly objective and not influenced by staining process or interpretation variability. Recognizing the challenges in MASH patient management and leveraging the advantages of our SHG imaging and AI image analysis, we are bringing QFibrosis to routine clinical care as our newly launched LDT FibroSIGHT Plus. FibroSIGHT Plus is the commercial name for the clinical diagnostic service. The plus in the name highlights its quantitative nature, and FibroSIGHT highlights the clear visualization fibrosis, which underlies it all. Powered by QFibrosis, FibroSIGHT Plus brings automated quantitative analysis of liver fibrosis to routine patient care for more consistent patient evaluation, with reduced variability for more informed treatment decision-making. So working off the SHG image, QFibrosis automatically quantifies multiple fibrosis-related collagen architectural and morphological features detected by SHG and attributed to specific zones liver biopsy and translates them into a single value of fibrosis. The result is fibrosis expressed on a continuous scale rather than in discrete categories. As illustrated in these slides, different MASH biopsy samples were analyzed, and a QFibrosis value was generated, showing how QFibrosis scores track with increasing grades of fibrosis F0 to F4. QFibrosis values can easily be translated to ordinal categories corresponding to the NASH-CRN fibrosis system to offer clinical interpretability based on predetermined thresholds. FibroSIGHT Plus can thus provide reproducible and objective fibrosis assessment to support diagnostic accuracy and supports therapeutic decision-making. The QFibrosis model has been trained on conventional NASH-CRN stages through feature extraction and feature selection that best correlate with expert histopathologist fibrosis scores on hundreds of samples from F0 to F4. In the training dataset, the QFibrosis scores track very well with conventional MASH fibrosis staging with Spearman's correlation of 0.816 for QFibrosis values against the assigned NASH-CRN stages. The model achieved an ROC of 0.7716 in the training cohort, indicating good overall discrimination across fibrosis stages. This suggests that the Q-fibrosis score reliably increases with disease severity and supports its use as a continuous biomarker. When Q-fibrosis values are translated to ordinal stages based on thresholds compared to the assigned reference NASH-CRN stages, the model detected clinically significant fibrosis of stages, i.e. F2, 3, and 4, with 86.3% sensitivity and an accuracy of 80.1%. The excellent performance of Q-fibrosis model in external validation datasets demonstrated strong generalizability across independent cohorts, with Spearman correlation coefficients of 0.739 and 0.737 in 2 external validation datasets, and sensitivity of detection of clinically significant fibrosis as high as 89.7 and 91.1%. These results support the model's robustness and clinical relevance beyond the training population, particularly for identifying clinically significant fibrosis. Although digital pathology solutions offer improved performance, validation studies may only demonstrate accuracy of 80% to 85% due to the ceiling effect of the current imperfect gold standard. The entire FibroSIGHT Plus workflow, including SHG tissue scanning and AI-based image quantification, is highly reproducible across instruments, operators, and sample batches. Multiple SHG scans of the same tissue were obtained on multiple Genesis 200 scanners, and Q-fibrosis values were calculated, and the intrasystem repeatability rate was 93.77%, and reproducibility rate was 93.50% using median of Q-fibrosis values as the ground truth. When appreciating minor yet significant differences in fibrosis, amounts measured with quantitative sensitive approaches, such as FibroSIGHT Plus, high repeatability and reproducibility are paramount, and with these excellent reproducibility results, sensitive detection of clinically meaningful variation while minimizing subjective bias is assured. To conclude, in workflows where biopsies are required downstream of NITS, FibroSIGHT Plus enables the most accurate assessment of fibrosis, ensuring that the value of each biopsy is fully maximized. This approach prevents overtreatment, reduces misdiagnosis, and helps optimize the use of limited healthcare resources. Looking ahead, FibroSIGHT Plus is positioned to play a central role in enhancing the impact of biopsies within the clinical workflow. Coming up, we will be presenting new data on the performance of community pathologists and FibroSIGHT Plus in staging MASH fibrosis. In addition to FibroSIGHT Plus, we offer a comprehensive suite of liver pathology services, all delivered from the same integrated lab. This includes expert histopathologic evaluation and stain interpretation. By centralizing both traditional and AI-enhanced workflows, we streamline turnaround times and maintain rigorous quality standards across every specimen. To learn how to get started with FibroSIGHT Plus or follow us on LinkedIn, simply scan the QR codes on screen. Thank you.